And I will be your moderator for this webinar. I am currently a PhD candidate in the Integrated Biomedical Sciences program at UT San Antonio, and I'm currently conducting my doctoral research under the mentorship of Dr. Shivani Ruparel within the Center of Pain Therapeutics and Addiction Research, as well as the Department of Endodontics Pain Laboratory, where we're studying the mechanisms of oral cancer pain. So before we begin, just briefly going over how questions will be handled throughout today's session is that audience members can ask questions to the speakers and moderators using the Q&A function. You'll have the view option menu at the top of the window that allows within the zoom in and on shared in the screen overall. And for our recording for the webinar itself, that will be available on the painresearchforum.org website and as well as our YouTube channel. Then I believe, Becky, if you could proceed with the slide. We do have our journal club series that's continuing on. For our next session, it'll be on Tuesday, June 16th, 2026, 10 a.m. London time, 5 a.m. Eastern time. And that'll have our featured guest here with Dr. Karen and Dr. O'Connell speaking on this paper, Identifying Social Factors that Stratify Health Opportunities and Outcomes in Pain Research. Consensus recommendations for the collection and reporting of equity-relevant data. So tune into that and please register for the event. There's a QR code here on the screen. So now, without further ado, I have the pleasure of introducing our first speaker, which is Dr. Armen Akopian. So Armen, if you could kindly share your slides with us as I introduce you. So Dr. Akopian has a multidisciplinary scientific background that spans over theoretical physics, biophysics, molecular neuroscience, as well as pain research. He completed his PhD training in molecular neuroscience under the mentorship of Professor Georgiev, who was a twice nominee for the Nobel Prize. And he followed up with his postdoctoral training in pain research at the University College of London under the mentorship of Professor John Wood. Dr. Akopian received specialized training in electrophysiology during a sabbatical at New York University and in behavioral physiology at UT San Antonio, where there he became faculty in 2009 as an assistant professor and eventually promoted to full professor in 2019. So for Dr. Akopian's research, his main topic, it really looks at the mechanisms of underlying pain across several chronic pain conditions with an emphasis on pain affecting the head and neck region. So we are very pleased to have Dr. Akopian with us today, and now I will turn over the webinar to him. Thank you, Armen.
Thank you for introduction. Good morning, everyone. And thank you for invitation for the presentation. Traditionally, molecular mechanism for pain is studied in the models, and this is a legitimate approach, but several questions, especially heterogeneity in real deal clinical conditions, very difficult to model, and maybe in some extent it's impossible. And this presentation will be to understand how this heterogeneity can be assigned to some specific molecular signatures. Why it doesn't switch? Okay, so temporomandibular disorder, there are two types, arthrogenic and myogenic, and each one of them have got subtypes. For example, temporomandibular joint disorder can be osteoarthritis-based, rheumatoid arthritis, or idiopathic. In the meantime, myogenic can be divided into two types. One is local myalgia and another myofascial pain with referred. We will talk mainly about myogenic TMD. Main also property of this condition, what it has got the predominantly in females, in our hands it's a 1 to 9, high comorbidity with other pain conditions, and treatment extremely challenging. There is no clear-cut pharmacological approaches, and it usually used different approaches such as muscle relaxant and physiotherapy, for example, and sometimes even Botox. Now about mechanism. Mechanism for myogenic TMD, it's not known, but there is a presumption what it's start from muscle overload which lead to ischemia, and ischemia lead to inflammation, and inflammation actually control all pain pathway through peripheral mechanisms. So pipeline for our clinical studies, which is mainly conducted in the Karolinska Institute. Part of this clinical domain is located in Karolinska Institute under supervision of Malin Ehrenberg. So we first do pre-screen, and after collect the questionnaires, which is just several of them listed below. Then we get in consent and QST, and finally we'll get in samples of muscles. Now, samples of muscle, they are very small, and they are usually 1-3 cubic millimeter. Therefore, approaches which you see for processing these samples very challenging, and we will stop all them for each one of them, how to avoid possible mishaps during analysis. Now, most critical part of any clinical research is your inclusion and exclusion criteria. There is no right and wrong here. It depends on what we actually would like to achieve. In our case, we got it's very strict conditions. We worked only on females because males will not provide enough power. And we excluded many conditions such as like dental pain, neuropathic pain. Only condition which we accumulated separate group is a widespread pain condition such as fibromyalgia. And you can see on the left, we started with 1,332 participants, and eventually samples were generated from around like 60 patients. And beside that, we also generated from healthy control around 22 samples. Now, how we collect the sample? There are many ways to collect samples. Usually people generate one sample and that's it. We avoided to do that, and every patient provided 4 samples, and 4 samples internal control from tibial muscle. And on top of that, because the study will be used for clinical actually trials with the Botox on the saline, it played a placebo effect, and we collected samples in the 1 month, the 3 months after injecting saline. Now, let's start from bulk RNA sequencing, which was a major driving approach to understand what's going on. One way to analyze it is just to analyze against internal control, which is a tibial muscle. Another way just to analyze all naives against all myofascial pain or local myalgia. Neither of these approaches produced actually satisfactory results for several reasons. Second approach had such a heterogeneity, we almost got to nothing. And the first approach, based on presumption, with muscle and tibial muscle, they are the same. They are similar, but they are not the same. So what we done, we've decided to get clusters from 2 of these conditions, and clean cluster for naive, healthy control, and perform several comparisons depends on this cluster. Now, so we use the usual principal component analysis, and you can see what many samples, which are in green, also blue and pink, they have been excluded. Now, all samples in the red, this area, they are double. If they are not double, they've been excluded as well. That's a very important to keep a double. That will ensure what your actually healthy control is homogeneous, not heterogeneous. So if we look on the local myalgia on the left and myofascial pain with referral on the right, you can see we got the clusters. Now, if we look at these 2 clusters, they've got only singular probe. They've been excluded. And here, if we noted what these samples had a salivary gland, they've been excluded as well. Now, question is, we will say these samples, which is some of them dual, that was a requirement in every cluster to have a dual samples, which were generated from the same patients. We presume what they should be absolutely the same and not affected by some processing during bulk RNA sequencing, which contain many steps. So we shuffle this blue one into 5th or 2nd, and the same here, 1 in 3rd. Now, what happened after we analyzed gene plasticity? You can see what they are quite different. One for local myalgia is got inflammation upregulated, and we will say, and another one is got almost no upregulated genes. Inflammation actually was downregulated compared to health control, and all processes, vascular, muscle structure, neurogenesis, gliogenesis, they all went down. The same could be observed for myofascial pain with referral. All these inflammatory processes also went down, and upregulated genes was almost absent. Okay, now we have got data on how long these patients experience this condition. Based on this, we realized what that samples, which has got inflammation, was, we will say, in early stages of chronic pain. You can realize what this is all chronic pain because earlier samples, which we had, these patients already had 2 years myalgia. Okay, this is an all chronic condition. It's a just chronic condition has got stages. You can see if, and what the stages difference is what inflammation actually, for we'll say, goes down gradually, and muscle degeneration and especially extracellular matrix degeneration goes, we will say, towards descent as well. Now, what we done, we ask AI to look on, we will say, these samples, which is the differentially regulated genes, and actually tells which type of genes correlate with differences for both local myalgia, MYA, and myofascial pain. And this is what AI generated for us. This is the 3 top genes: TGF beta 2, GDF15, and TWIC. Now, you can see what TGF beta 2 mainly was observed in local myalgia is sometime in the myofascial pain. And that's what we'll say also show at the point where in the patients this gene regulation disappeared. And if, some people who don't know what is a TWIC belong to TNF superfamily. It's a TNF superfamily 12. And there's a got receptor, which is a TNF superfamily receptor 12A. I think receptor name, another alternative is FN1. So now single cell sequencing we run to understand what type of cells express actually these genes. Since we can generate only 500, 600, like 1,500 cells, direct, we will say, single cell approach doesn't work. What we done, we mixed with mouse TG, and then we will say run together in just software separated with human cells from mouse cells. Otherwise, we will say this is the only way to approach this. So every, we will say, sequence from patients usually generate around maximum 700 cells. And these are what we got. I mean, I will not, we will say, focus on what type of, we will say, cluster they are. The major, we will say, point which is needed to be made, what TWIC is expressed in the fibroblast. This fibroblast they name FAP, we will say, a type of fibroblast which can turn into the fat cells, or they can proceed in a control, we will say, muscle progenitor cells. And in a smooth muscle, the same goes for all these genes, if you look. Now, next step, what we've done is this is the only step which we use the animals for our experiment. What this FMO, it's usually mimic, we will say, step when you overload muscle. You stretch mouse open. We've done only 10 minutes to be absolutely sure what we maximally will say, mimic clinical condition. And then we injected TGF beta 2. You can see it at 25 nanogram, 10 nanogram, and 10 nanogram. And this measurement here shows on this 35 days. In the reality, it's lasts for literally forever. At least we measured until 90 days. And interestingly, also this picture doesn't show if we use a TGF beta antagonist after day 30, it's useless. Basically, it can't stop. What it tells, what also like in a patient, probably this molecule responsible for transition into from a one chronic phase stage to another. So what we suggest schematically, what is happening, we'll say after overload, acute pain transit to the chronic pain, which we obviously don't see in the patient because earliest is a 2 year. And then we will say in the muscle, you have got two mechanisms. One is trying to drive to the next stage of chronic pain, and another one is attempting to restore tissue injury. And eventually these genes probably take over and transit into the, we will say, latest stages of chronic pain conditions, which we name persistent. I mean, it can be labeled any way you want, but basically they are second phase, and minimum, we will say, years which are required to get this persistent condition. It's around 7-8 years. So I would like to say thank you all my colleagues on the rejoined, which participated in this study, and also for many students in my lab who actually perform this project. One last thing was we usually use the model for myalgia in the mice, and if you compare what's happened in humans and the models which is traditionally used, they have got no any overlap, or this overlap so minimal. What it's question arises why we needed to study animal model, which maybe don't represent reality, which we observed in the humans. Thank you.
Thank you, Armen. Wonderful work. Very exciting to see. And then I thought it was very fascinating that you're using AI to find your DEGs of interest. So it's the power of technology.
Yes, this we will say approach which develop our colleagues in the computer engineering department UTSA can be used not only for parameters like how long people experience this condition, but also for different pain questionnaires. I mean, we asking question not only VAS difference, we only VAS, which is a quite. We also have got QST data and different questionnaires which represent, we will say, acute feeling of pain. It's during 2 days, and there are the longer when they experience frequency during 2 weeks. Okay,
Mm,
and this technically we will say this software can be run. Unfortunately, haven't been published as yet. Can be find the correlation between molecule gene regulation, or we will say cell plasticity, and any type of questionnaires which we'll collect from patients.
wonderful. Thank you so much, Armen, for the information. So now on to our second speaker. I have the pleasure to introduce Dr. Joshua Emrick. Dr. Emrick, if you could please share your slides as I introduce you. Dr. Joshua Emrick is an assistant professor in the Department of Biologic and Materials Sciences and Prosthodontics at the University of Michigan School of Dentistry. He was awarded his DDS and PhD in oral and craniofacial biology from the University of California, San Francisco School of Dentistry, completing his dissertation with Dr. David Julius, who won the Nobel Prize in 2021. And prior to joining Michigan, Dr. Emrick conducted his postdoctoral studies as a dental clinical research fellow at the NIDCR with Dr. Nicholas Ryba. Dr. Emrick is a Biological Sciences scholar, and his lab's work is supported by a NEOMS UC2 as a member of the Rejoin Consortium and a NIDCR R01. And he has a plethora of awards that I will list here. He is a Calvi Research Fellow, member of the Omicron Kappa Ellipsin, and is the past recipient of the Distinguished Faculty Research Mentor Award at the NIH Office of the Director Award, UC Regents Scholarship, Robert W. Rule Award, Elizabeth Furman Gardner Award, Intramural Loan Repayment Program Award from the NIH, Fellows Award for Research Excellence from the NIH, and last but not least, the AADR Hatton Award. So thank you once again, Dr. Emrick, for joining us in today's webinar, and now the floor is yours.
All right, thank you. Can everyone hear me okay? And can you see my slides all right?
Yes.
Okay, all right. So we're going to change gears a bit. So Armen, that was a nice talk. Thanks for sharing your work on myalgia and temporomandibular disorder pain. So we're going to talk about teeth and the innervation of teeth. And I think that, you know, this is relevant because I think that, you know, the innervation of the teeth, what we're going to talk about is how they serve the biology of an organism and then also contribute to fitness. And I think this is something that we didn't appreciate until we worked through this project. But I also think that this is relevant for sensory biology and also nociception more broadly, because what we're learning is that this type of neuron, this mechanonociceptor, typically we also call them HTMRs, it seems like they're playing a role throughout the body in generating reflex responses. And so there's a choose your own adventure part of this talk. So I'm going to let everyone sort of determine how we're going to talk about gain of function experiments that we performed. But I'm really excited to be here today. Thanks for the invitation. My only regret is that 12 minutes is not enough. So I'm going to do my best to do this justice. Okay, so I'm really interested in the somatosensory system. And here I have depicted the trigeminal portion of the somatosensory system. So these are the sensory neurons that are outside the brain and the spinal cord. They're in our peripheral nervous system. And their job is to send out their projections and detect the environment. Okay, and so that could be the external environment. It also could be the internal environment if we're thinking about interoception. The idea is that once these neurons sense something through their receptors. They relay these signals toward the brain, and that gives us the basis for our perceptions and our reflexes. And again, this is the trigeminal ganglion, and this is an illustration of that in the human. Primarily speaking about mouse models today. DRG are in the rest of the body. So if you ever go to Detroit Metro Airport, some of you might be coming there for the Rejoin Consortium meeting this year. You'll see there's a giant gumball machine, and I really like this as an illustration because it just shows us the different. In my mind, it's an illustration of the different types of sensory neurons that we have. And if you think of each gumball as a kind of neuron, and if they have the same color, they have a similar function. And the idea is that there's a lot of heterogeneity, even though I've depicted these as sort of all uniform in color, maybe different in size. But each of these neurons might belong to one or more classes, and we can ascribe function to those. So partly the way that we've done this is through the advent of single cell sequencing and also single nuclear sequencing. So this was work that was done in the Reba lab. And right when I was coming on, Lars von Buchholz, one of the staff scientists there, had just worked out a way to take the information that we had from transcriptomics and figure out how to apply that using in situ via machine learning to then designate neurons in sections. So here we have, again, the gumball machine, right? We have around, and this is an earlier sort of like visualization for how many different transcriptomic classes there are. Each of these dots is a neuron, and the way that they cluster in this t-SNE plot is just based on their similarity of expression, all right? And then if we perform a series of in situ hybridizations in vitro, we can actually predict the type of neuron that these go back to. So realistically, we have a way in vitro of ascribing identity and function using sort of a simple in situ hybridization assay. And this can give us information about the transcriptomic class, or what the transcriptomic class might be, and also there's relevance for function here as well. Okay, so my lab is interested in how this system contributes to oral function and perception. All right, so we'll be talking about the tooth today, but we have some other projects looking at the temporomandibular joint, and that's, I have the pleasure of collaborating with Armen as part of this Rejoin Consortium funded by the UC2 grant. And then also I've been looking at the innervation of the salivary gland. So I'm not going to talk about any of those other tissues today. I'm just going to talk about the tooth, and I'm not going to talk about all the work that we've done with the tooth. I'm just going to talk about this work that came out last year that was spearheaded by Liz Ronan, a postdoctoral fellow in my group. We're going to talk about the internal innervation of the tooth. This year, we had a really awesome collaboration with Bo Dwan. He's also at Michigan, where we looked at some of the external innervation around the tooth and how that contributes to gnawing and dopaminergic signaling, which actually rewards gnawing and actually helps maintain dentition. If you're interested in looking at that, I've got the link, some QR codes. But we're going to primarily talk about this top story, and I'm going to try to cover as much as I can with it in the time that we've got. Okay, so when we started out, the idea, and so I'm actually a practicing dentist. I do this very, very minimally because I'm primarily in the lab. But as a dentist, I deal with pain a lot, and patients also deal with pain. Unfortunately, that's one of the driving factors that brings them to the dentist. And so the idea is that the innervation of the tooth triggers pain. Okay, and that's in the context of damage and inflammation. Typically, sort of in our normal state, we're not feeling this so much. And this has been appreciated for decades. I mean, this is now more than 50 years ago. Okay, Barry Cecil and these authors, they were thinking about tooth innervation as a way to understand the pain system. All right, and they were asking whether or not basically opioid signaling and endogenous opioids and endorphins might be signaling in this pain pathway. But this is sort of to illustrate that this has been the predominant theory for a long time. Tooth innervation causes pain. Okay. So the idea is that that some of the innervation has been defined, but it's not so clear, at least within the context of transcriptomics, what types of neurons might be innervating the inner tooth. Okay. And so we wanted to try to understand, okay, beyond pain, what neurons, what could the function of these neurons be? And also we wanted to update our understanding of their transcriptional profile. What types of classes, if we interrogate them, what might these represent? And we had some ideas, and this is based on past literature, that we might see some expression of TRPV1, maybe. And that was thought to allow this innervation to sense heat and hot pain, calca and piezo2. So we had these were not transcriptomic or related to the transcriptomic data. This is just sort of one-off, either IHC or qPCR, probing these neurons to ask if there was expression. There's a really wide range of reports and percentages of what each of these neurons or how many might be expressing any of these markers. All right, so this is the team that makes the work happen. It's a real privilege to work in my lab and to work with all these brilliant folks. Liz is here. I bolded the folks who have contributed to this work. And so we have some current lab members and also some past lab members who've moved on. Not like thoroughly. They've gone to medical or dental school. And then I have to thank collaborators at NIH and within our school, and also the K-22 grant, which helps support some of this work, in addition to these T32s that supported Liz. Okay, so we're going to talk a little bit about what Liz and company have been thinking about. What we wanted to do was to ask, using in vivo calcium imaging, what might these neurons that are innervating the tooth be responding to? So the strategy here, and we didn't use TRPV1 as a driver line here, sorry about that typo, is that we would express GCAMP in some of the neurons of the trigeminal ganglion, and then we would monitor their activity upon oral stimulation. Okay, so the idea is that these are still frames of the surface of the trigeminal ganglion. If we actually, you can see in this preparation, we create a window to the TG. We remove the overlying cortical and midbrain structures, and then we can actually visualize the surface directly. That's what we're looking at here. And on baseline, these neurons are fairly quiet, at least based on the calcium imaging. And then when we provide an oral stimulus, you can see that these cells are lighting up. And this fluorescence is quite nice, and it's transient. This is just a still frame of this, but we can get the idea of hundreds of neurons firing simultaneously using this approach. All right, but we didn't want to see all the neurons. We wanted to see the neurons that innervate the tooth. And so what we used was a strategy of applying voltage pulses to the tooth. So we used 0.6 hertz, and we applied about a 4-volt potential to the tooth, and then we visualized the surface of the ganglion. You can see that we recruited, so this is actually process data just to bring out the signal. You can see that we actually saw this sort of pulsing effect. These are the neurons that are entrained to the stimulating frequency and the voltage. So that was pretty exciting. We thought we had a way of, without damaging the tooth, applying this voltage and identifying neurons of interest. So after identifying those, we performed this, multiple iterations of the experiment. And what we found is that we could stimulate either the first molar or the second molar. There were actually dedicated separate populations that were responding to each of these pulses. So we've got this pseudo color down here and concatenated. You can see that these are separate populations. You can also see, so this would represent fluorescence here on the y-axis. This heat map also represents our fluorescence intensity. And you can see that there's two different patterns that come out. One is that depending on which tooth you are stimulating, you have alternating populations firing. And then you also have the striated pattern, and that reflects our the 0.6 hertz stimulating frequency for voltage. So we find about 16 neurons in the prep, and these are fairly large neurons as well. Okay, what we did afterwards, following this stimulation, is we removed the ganglion, performed whole mount in situ hybridization, and then we aligned our imaging data for our fluorescence with the in situ, and we found that these neurons, maybe not to our surprise, we've done some other experiments, but these neurons were likely to be very large diameter neurons, and they also seem to be nociceptors based on their expression of STN10A. So this is a little bit different than what we predicted based on some of prior literature, but it was interesting that we've identified a population of myelinated nociceptors that seem to be innervating the inside of the dentin. Okay, so we wanted to check that. We actually used two different Cree drivers and sparse labeling strategy using AAV9. We actually injected this directly into either the pup or the TG, and we found that we could achieve sparse labeling of these fibers that are actually penetrating into the dentin. I think I have a tooth here. Okay, so this is a tooth structure. You have the inner pulp, the dentin, and the enamel. So what we're actually looking at here is the border of the dentin and the pulp. And you can see that these fibers, we knew that the anatomy of that the dental innervation should penetrate to these structures. But what we wanted to ask was, were these populations that marked by S100B and SCN10A actually penetrating the dentin? And we were able to visualize this quite nicely. This was actually some of the first evidence that indicates that the receptive fields seem to be overlapping for these neurons. So what we wanted to ask next was, do these neurons detect damage in real time? And so what we found is after identifying them using the electrical stimulation, if we used a dental burr to actually shave the surface of the tooth off, we found that if we if we scrape the surface superficially, we saw that there was some response. And if we cut into the enamel, we saw that this response was exaggerated. So we found that additional neurons began to respond when we cut the tooth more vigorously, and then also that their response intensity increased as well. So it gave us a sense that this population actually tells us that when the tooth is being damaged, and the more that we're damaging it, the more neurons that are recruited based on population and activity. This is very minimal damage. I just wanted to show this because even if we are cutting into the enamel, this is actually very little damage. You can see this sort of fine dust. This is just the enamel being shaved off. So we're not drilling, so deeply into the tooth. It's actually fairly superficially. And we did some control experiments, which I'm not showing, that neither vibration nor heating actually produced these same responses. So we do think it is actually the damaging component of the tooth. Okay, so the last thing I want to talk about is some of the gain of function experiments. So we wanted to ask what the consequence of activating these neurons are. So I'm trying the Slido out. If we can quickly give me any indication, if you'd rather hear about chemogenetics or optogenetics, we can go forward with the next part of the presentation. Chemogenetics, we'd be thinking about how we've used lab gym. Okay, we got one person. Lab gym that allowing us to enable the evaluation of orofacial pain, or optogenetics, we actually discovered a motor reflex that's a consequence of activating these neurons acutely. We've got one. Anybody else? Three? Okay. I'm going to wait till we get to 20. No, I'm going to wait till we get to 10 because we're running out of time here. It's close, neck and neck. Okay. Motor reflex. I'm going to be a tiebreaker if we don't get one more. All right. I'm going to go to optogenetics, okay? All right, so I'm going to skip all the chemogenetics and lab gym that we used. All right, well, what we wanted to ask was, if we were to optogenetically stimulate these neurons, what might happen? And we knew that there are trigeminal reflexes. All right, so what we reasoned was that it's possible that activation of this innervation might contribute to one or more of these trigeminal reflexes. So we actually used ST-antennae Cre, which is, I showed you that these neurons express to ST-antennae. We used this Cre line to drive channel rhodopsin. And we actually applied an optical fiber directly to the tooth. And you'll see that when we turn on blue light and we stimulate, we actually get a recoil of the mandible here. So this mouse is under anesthesia, okay? And when we apply this fiber to the tooth and we stimulate the endings directly in dentin, that we have a recoil of the mandible here. All right, so we performed a series of experiments, one just to get an idea of the time scale. This is a very rapid reflex. It happens within 15 milliseconds. All right, it's fairly reproducible. So if we give trains of stimulation, we're able to elicit this mandibular deflection over and over again. It's on the order of hundreds of microns, at least within this paradigm that we're using. And if we use a green light, we are not able to evoke this. We're also not able to evoke this response if we stimulate other oral tissues other than the tooth itself. And one thing to include is that we actually used a paradigm to transduce neurons directly from the tooth, all right, and then stimulate the soma. And when we did that, we weren't able to elicit the full-on muscle contraction and the mandibular movement. But what we did find is that we had EMG activity in the anterior digastric. So these neurons, when they're stimulated directly, their somas are able to drive this some muscle activity. We reasoned that if we had higher transduction efficiency, or if we were able to activate this population in total, we'd probably be able to elicit, or I mean, we may not. There might be other muscles involved as well. But this is really exciting because it gave us the basis for how this reflex is initiated. Okay, the only last thing I want to say is that this is work, a preprint that came out of David Ginty's lab, led by Karina Lezgieva. And the idea here is that when they're activating these HTMR populations, which are similar to those that are innervating the tooth, they're able to find that they have withdrawal reflexes in the paw as well. So it seems like this is a logic for sensory biology throughout the organism, and not just in the trigeminal system. It's really exciting that this work was reproduced in another part of the system. Okay, so I'm going to skip the summary. The only thing I would say is that realistically, the implication here is that this might be a population that's responsible for telling us if the tooth is damaged, like in a cracked tooth, or it also might be, and certainly it also represents the population that's being activated by the dental burr. So when we get dental anesthesia, we're likely inhibiting the signals from this type of population that would be in the human. As I told you, the mouse tooth, when we're drilling it, it triggers a response to these neurons. We know that if we go to the clinic and we treat patients without anesthesia, they feel that quite severely. So we think that there's probably an analogous neuron in human tooth as well. And we think that this is also a potential candidate for what might be causing pain during pulpitis as well. So this is an attractive neuronal population that we might inhibit to help us to address dental pain as well. Okay. And then this is an idea of, the last thing is this actually gives us a basis for why we need a crown after we have root canal therapy. So if you remove the inside of the tooth and you remove the innervation, you no longer have this protective response. All right. And that's actually what's helping us prevent fracture of our teeth. And so the longstanding idea was that we weaken the tooth during root canal, but realistically, if we remove the innervation that's providing this protection and this reflex, then it's actually once it's lost, it actually contributes to fracture of these teeth. And so this is why we need to crown teeth, especially in the posterior following root canal treatment. All right, so with that, I'm a little over. Tried not to. Dr. Pellesi, please forgive me for going over.
Thank you so much, Dr. Emrick. Yes, just to remind everyone, we have a few questions coming in the Zoom Q&A. So just for the interest of time, we're going to go ahead with Dr. Pellesi's presentation, and then we'll address the questions at the end. And I see a few had their hands up as well. If you could just direct those questions to the Q&A, we will get to those. All right, thank you so much. So now to our final speaker. It's my pleasure to introduce Dr. Pellesi. So Dr. Pellesi, if you could share your slides as I introduce you as well. So
Yeah.
Yes, Dr. Lanfranco Pellesi is an assistant professor at the University of Southern Denmark, where he conducts translational research on migraine and headache disorders to identify novel therapeutic targets. So he is an author of more than 70 peer-reviewed articles and principal investigator of human provocation trials in individuals with migraine. He has been a part of research teams showing the role of VIP in migraine induction and KATP channels in cluster headache. Again, we are grateful to have you participate today, and now I will turn the session over to you. Thank you.
Thank you very much for the invitation. And can you hear me?
Yes, loud and clear.
And can you see the screen?
Yes.
Great. So today I will talk about these experiments that are called human provocation models of headaches in general, and I will have a focus on the migraine disorder. Before talking about my own work, I think it's wise to talk about what it is, this provocation model. So what does it mean, provoking headache or migraine in a human being, and what we have learned from it. And then I will try to illustrate my own work, our studies at the University of Southern Denmark, with some glimpse also about our future trials. So, a human provocation model of headaches in general is a way to test a potential substance, might be a drug, for example, might be a peptide. So, in general, is a compound with a very specific mechanism of action, with the idea that if we are testing a mechanism of action that is close to the headache pathophysiology, we will have a quite high rate of headache induction. When we are testing specific disorders like migraine or cluster headache, then the idea is that we will have a high rate of or migraine of or cluster headache. These trials are done essentially in order to understand the pathophysiology of headache disorder and to have at the end potential therapeutic targets. So switching now to the migraine field. So how a migraine experiment is conducted. So essentially we have a screening at the beginning where we evaluate patients and his or her migraine. When the patients decide to participate in the trial, we allocate the treatment. We call it treatment, but it's actually a trigger. This trigger can be an oral drug, for example, but there might be also injections of peptides involved. These trials, I forgot to tell you that these trials are typically randomized, placebo-controlled crossover trials in order to get the best scientific evidence from these trials. And patients, they act as their own control. So meaning that one day they will get the trigger and the other day they will get placebo. They generally stay on site with a monitoring period of approximately 90 minutes, and then the window for headache is actually lasts around 12 hours post administration. The primary outcome is typically the difference in incidence of headache between the trigger and placebo treated groups. In the case of migraine, we have validated migraine criteria in order to define an experimental migraine attack provoked during these trials. These experiments are not new. These experiments have been done for several decades. Very briefly, we have discovered several compounds able to trigger migraine in people with migraine at a high rate. For example, some peptides like CGRP or PAC38, but also some drugs with a specific mechanism of action like sildenafil, cilostazol, and lastly, leucromacalin, that is a potassium channel opener. So in order to have a sort of a unifying hypothesis about these provocation trials, a paper on the New England Journal of Medicine has been published on a few years ago, showing that the responsible for all of these painful information coming from these trials in the context of migraine are the trigeminal ganglion neurons, and in particular the projection of these neurons that are at the trigeminovascular level, so the projections that are in close proximity to intracranial and extracranial blood vessels. The signaling pathways that have been discovered through these provocation trials have been summarized in this picture, where we see avascular smooth muscle cells of it can be of an intracranial or an extracranial artery, for example. And then we see that essentially we can define some CIMP-related pathways where we have some peptides like CGRP and PACAP that act on this. And then we have also CGMP signaling pathway possibly involved. The idea is that all of these pathways converge on these potassium channel openers with the overall final results of having an efflux of potassium that can sensitize the perivascular sensory afferents that are very close to the cranial arteries. These experiments are not just experiments for discovering new signaling pathways. They have already they have brought us some concrete treatments, and they are possibly bringing us new treatments. So I just brought here. I'm just showing you a couple of examples. So for example, the CGRP peptide has been one of the first to be tested in this setting, and has been the results of a provocation trials with CGRP have been supportive for the development of several drugs now that we have on the market. So for migraine prevention and migraine treatment in the acute setting, we have now different molecules. And more recently, we have an anti-PACAP peptide that is now going into the phase 3 trial. with the positive results from the phase 2 trials. In that case, provocation trials, I would say they have had a much more prominent role in order to guide the development of these drugs. So essentially, what we started to think at the University of Southern Denmark, and then what has started our experiments was just an easy consideration, I would say. So, for example, that migraine is essentially a female disorder, especially after puberty. Unfortunately, the provocation trials that I have shown you and the signaling pathways that have been built up according to these studies were essentially coming from studies conducted in females. So in every of these studies, the population of females with migraine was between the 80% and 100%. So we started last year a study with sildenafil. Sildenafil is an inhibitor of vascular phosphodiesterase. Its main mechanism of action is responsible for the treatment of erectile dysfunction, but is also supposed to cause headache in people with migraine. So we did a randomized placebo-controlled crossover study in men with migraine, the first of its kind, and then we tried to replicate previous trials showing a high rate of migraine-like induction in women with migraine. What we have found essentially is a similar percentage, high rate and high percentage of migraine-like attacks in women after the intake of sildenafil that was taken orally, 100 milligram. At the same time, we have shown that the migraine-like attacks were not so frequent in men with migraine after sildenafil compared with placebo. Headache was common in men after sildenafil, but not. This headache did not fulfill the migraine-like criteria that we used. For this kind of experiments. The reasons behind that might be different. So we think that there might be an hormonal modulation going on in women with migraine, potentially driven by estrogens and prolactin. There might also be a pharmacokinetic differences between men and women. We used the same dose, 100 milligram of sildenafil, but we did not measure the sildenafil drug concentration. So we do not know if a pharmacokinetic differences drove the difference between men and women, and there might be also a different symptom expression. So, for example, men are reporting quite constantly fewer associated symptoms to headache. So the idea is that we might have seen still migraine in men with migraine, but because they report less accompanying symptoms, their feature, they do not fulfill the criteria for a migraine-like attack. So now we are doing the specular study, so with on the CMP related pathway using cilostazol. We have just finished the experiments. So we have done essentially the same study, but instead of testing the CGMP signaling pathway, now we have tested the CIMP related pathway. So the pathway that is more related to the peptides, where we have some therapeutic targets. The idea that we have is that if we see some differences in these CIMP and CGMP mediated pathways, there might be potential for changing the diagnostic criteria between men and women with migraine, biomarkers as well, and also future treatments that might be more selective, and then targeting only females or men with migraine according to the signaling pathway that is more prevalent on the sex of these patients. In general, I would say that every kind of difference might have a role in every aspect of migraine research, where we tend to think that men and women have the same pathophysiology. They share the same mechanisms of the disorder, but maybe that is not the case. Now, at our lab, we are focusing on new therapeutic targets. So what we are trying, what we will test in this autumn is a histamine H3 receptor modulator that is called pitolisant in a cohort of women with migraine. The idea is that to revive a bit the link between histamine and migraine. So we know that the histamine H1 and the H2 receptor antagonists are not so effective for migraine, but now we have a drug that can modulate very selectively this receptor and might show potentially some migraine induction in these patients. And at the same time, we are also trying to test a drug that is called apremilast, used in arthritis, that is an inhibitor of phosphodiesterase 4. Compared to the previous drugs that I've shown you, like cilostazol and sildenafil, who are inhibitors of vascular phosphodiesterases. Apremilast is an inhibitor of neuronal and immune cells phosphodiesterase, in this case, the phosphodiesterase 4. So the idea is that we might show that there might be different cells where we can start to provoke migraine. And lastly, I have to thank, of course, our funding to get us the money to do these experiments, and our collaborators, my team, the students, the different students from the pharmacy degree and the medical degree, who have supported very carefully and very thoroughly our projects, and of course every participant who has decided to maybe to get a migraine or a headache attack during these experiments. And thanks for having me to present this. I think it's a very interesting and exciting research, and hopefully there will be some new advancements from these trials.
Wonderful. Thank you so much, Dr. Pellesi. So we have a couple of questions coming in with a Q&A. So I'll start. I think this one was directed to Armen. So TMD patients typically do not present with ongoing pain, but rather TMD is diagnosed by pain evoked either by palpation or jaw movement. So did you see any expression level on mechanosensitive ion channels in your work? Or mechanosensitive ion channels?
No, we analyze muscle. I mean, for the chance to get a neuronal. I mean, it's there is a chance if they are in a fibers on a very high level regulated. Actually, we do get some molecules which belong to axons, but they usually molecules which are responsible for axiogenesis and synaptogenesis. So we recently, what we done, it's actually done was in animals from dural matter, which is important for migraine. We got the same many genes which are regulated in axons. We just treated it, and broke it to single cell suspension and sequence again, and noted what all these genes disappeared, because by treatment like making single suspension, you probably rid of all axons. But I should be honest, but we did not specifically saw any channels in that one, at least not popular ones.
Okay, great. So for Dr. Emrick, someone asked if you are suggesting there is no somatic topic organization within the TG?
Yeah, I think that, no, I wasn't trying to suggest that, David. I think that I'm not sure what part of the presentation, that I accidentally made it seem that way. But what I would say is that there certainly are divisions to the trigeminal ganglion. I would say that the boundaries between those divisions are a little bit looser than they've been defined in textbooks. But certainly we have a very stereotyped localization of neurons innervating the mandibular structure versus the maxillary structures, and our imaging allows us to see some of the ganglion. And that is one of the limitations, is that if the neurons are buried. then we're not able to visualize those and their activity and their response profiles. But in this case, when we were looking at the mandibular molars, which is primarily what we were visualizing when I was talking about the in vivo calcium imaging, those are visible to some extent on the surface. We think some may be buried as well. We have labeling data which indicates time and again the relative localization of neurons innervating both the maxillary molars and the mandibular molars. It's stereotyped, but it's not hard and fast. I think there is some stochastic nature to the way that the ganglion develops. We haven't dived down into that as far as developmental biology goes.
Great. Now for Dr. Pellesi, Michael Gold had asked, he apologized for a second, saying he's sorry if he missed a part of the argument, but how does a potassium channel opener lead to the sensitization of an afferent terminal? The channel should hyperpolarize neurons and decrease excitability.
Yeah, thanks, Michael, for the question. I think you are right. If you look at the membrane of the sensory neurons that is close to the vessel, but instead here we are talking about another channel. So here we are talking about a channel on the vascular smooth muscle cells. So this is open, then there is an efflux of potassium, and the potassium that is going that is moved out from that cell is able to sensitize the neuron that is close but is not involved in any opening of any potassium channel. So I think this is a very good question, and it's a sort of a it's a sort of a misunderstanding, I think, for also for us who are doing this. Anyway, this is more for a preclinical part. What I'm doing is mostly essentially clinical, so maybe there might be some more detailed explanations about it.
And Dr. Pellesi, was there a sex difference in the delay to the migraine induced by the sildenafil or sildenafil?
No.
No?
Okay. No sex difference.
Okay. For Dr. Emrick, an anonymous attendee said you had a great talk, and they were wondering if you have analyzed the brain stems and found any similar related activation.
Yeah, that's a great question. Thanks, anonymous attendee. You don't want to be attributed to a compliment. In this iteration right now, I didn't include any of these data, but we have looked at a few different things in the brainstem actually, and it's ongoing work, and we are trying to write this up for preprint. We hope by the end of the year. But what I can say is that certainly with the opsogenic activation of the intradental neurons at the tooth, at a time point, I think it's around half an hour, we could euthanize the animal, harvest the brains, and we could actually look at CFOS expression. We found that corresponds to a few targets of the innervation, which we've also visualized. So it's really interesting. The brainstem projections of these neurons, it's not what we expected to some degree. On one hand, it's what we expected in that it does project to some of this, or at least the spinal trigeminal nucleus. But we actually found a couple additional targets, and we think that that's actually related to the sensoromotor loop that we think we've uncovered here. So really excited to come back next year for the PRF follow-up seminar and talk about that work. But it's a good question.
Good. Sounds like a plan. Planning a year in advance.
Okay, great.
Yes. And then lastly, this question from Michael Gold, I think it's towards you, Dr. Pellesi, that what is your thinking behind going after H3 receptors, and what do you think they're acting upon or acting on?
Yeah, so essentially the H3 receptors are autoreceptors, so at the presynaptic level, they are quite widely expressed in the central nervous system. They regulate the histamine release, so essentially the idea is that using this drug that is used for the treatment of narcolepsy and that is giving a boost in the histamine neurotransmission, the idea is that we might see more headache, in particular in relation to migraine, if there is a link between histamine and migraine. The idea is also to try this because I think it can be even more relevant in cluster headache that was in the old times called an histaminic cephalalgia. In clinical trials, pitolisant has shown a quite high rate of headache as a side effect in between 30 and 35% of individuals. So we think that it's worth a try, and then we will see if the results will be positive or negative.
Great. Well, those are all the questions that I see here in the Q&A. We had great timing, so it's always appreciated. So thank you to everyone, especially for our panelists, our speakers, for sharing their time and expertise for today, and for everyone that had joined us today for this Pain Research Forum webinar. I hope the discussion continues on and we get meaningful support for our webinars here and also conversations about the research in orofacial pain. Thank you so much.
Thanks again.
Thank you.
Thank you. Bye.